Number 1. Fungal parasitism. The zombie fungus. Picture a carpenter ant in a Thai rainforest. It has abandoned its colony. It is climbing a plant stem, not because it wants to, but because something inside it is pulling the strings. At precisely 25 to 30 centimeters above the forest floor, the ant clamps its jaws onto the underside of a leaf vein. The bite is so powerful that the mandibular muscles atrophy and lock. The ant will never let go. Within hours, a slender orange stalk erupts from the back of its head, releasing millions of spores onto the forest floor below. The organism responsible is called Ophiocordyceps unilateralis. It was first described by Alfred Russel Wallace in 1859. It infects carpenter ants from the tribe Campanotini and only carpenter ants from the tribe Campanotini. It cannot complete its life cycle in any other host. The infection begins when a spore lands on the ant's exoskeleton. A structure called an opressorium forms, essentially a biological drill bit, and it bores through the cuticle using a combination of mechanical pressure and digestive enzymes. Over 4 to 10 days, the fungus hijacks the ant's central nervous system. The death grip is not random. Studies in Thai forests have found that infected ants bite within a narrow window around solar noon, when temperature and humidity at that precise height are optimal for spore dispersal. The fungus can tell time. And it has been doing this for at least 48 million years. In 2010, a research team led by David Hughes at the University of Exeter, Conrad Labandera at the Smithsonian, and Torsten Wappler at the Steinmann Institute published a study in Biology Letters describing a leaf fossil from the Messel pit in Hesse, Germany. The leaf bore dumbbell-shaped bite scars identical to those left by modern zombie ants. It was the oldest known evidence of parasitic behavioral manipulation in the fossil record, but the timeline has since been pushed much deeper. In 2025, a joint study from the Natural History Museum and the Nanjing Institute of Geology and Paleontology described Paleophiocordyceps, a new genus preserved in mid-Cretaceous Burmese amber, roughly 100 million years old. Molecular clock estimates suggest the broader Ophiocordyceps lineage diverged from its relatives over 130 million years ago during the early Cretaceous. This is a parasitic strategy older than the extinction of the dinosaurs. Over 200 species of Ophiocordyceps have now been described, each one exclusively host-specific. A species that parasitizes one ant genus will fail entirely in a closely related genus. The ants have not taken this lying down. Colonies have evolved collective immune responses. Workers carry infected individuals away from the nest before the fungus can sporulate. Dead ants cluster in so-called graveyards at the colony perimeter, 20 to 30 meters out, never inside. In response, some Ophiocordyceps lineages have evolved to suppress early behavioral symptoms, hiding the infection longer before triggering the death grip. And there is a third layer. Other fungi, called hyperparasites, colonize the Ophiocordyceps fruiting bodies and sterilize them before they can release spores. It is a 3-tier parasitic arms race running across deep time. How the fungus actually controls the ant is still debated. The mainstream view, supported by Carissa de Becker's metabolomics research, is that Ophiocordyceps secretes specific metabolites that manipulate muscle tissue directly. The fungus does not control the brain. It controls the body. An alternative view holds that the fungus builds networks of cells around muscle fibers that physically contract the mandibles, creating a biological puppet that bypasses the nervous system entirely. And then there is the speculative question that nobody can quite dismiss. Could something like this ever jump to humans? In 2019, Arturo Casadevall and colleagues published a paper in MBU proposing that rising global temperatures are enabling fungi to adapt to mammalian body temperatures. The thermal barrier that has historically protected warm-blooded animals from fungal infection may be eroding. This is not science fiction. On October 25, 2022, the World Health Organization published its first ever fungal priority pathogens list, identifying 19 fungi as critical threats to human health. It was the first time fungal infections had ever been formally prioritized as a global health emergency. At the top of the critical list sits Candida auris, a pathogen that did not exist in the medical literature before 2009. It has since spread to every continent. It resists multiple antifungal drugs. Its mortality rate in bloodstream infections ranges from 29 to 62%. and it may have emerged precisely because of climate change. Meanwhile, valley fever, caused by the soil fungus Coccidioides, is surging across the American Southwest as drought and heat expand its range. A prehistoric organism thriving in modern conditions, fungi are among the oldest parasites on Earth. Their behavioral manipulation strategies are at least 48 to 100 million years old. And climate change is now unlocking new fungal threats to humans that medicine spent decades ignoring. But the next disease on this list does not manipulate behavior. It hides inside your heart for decades. Number 2, Chagas Disease, the silent killer. The Chinchorro people lived along the coast of what is now Chile and Peru. They are best known for producing the oldest artificial mummies on Earth, predating Egyptian mummification by roughly 2,000 years. The Atacama Desert, one of the driest places on the planet, also naturally preserved thousands of bodies across millennia. In 2004, a study led by Arthur Aufderheyer, published in the Proceedings of the National Academy of Sciences, tested soft tissue specimens from 283 of these naturally desiccated mummies. The bodies spanned 11 major cultural groups across approximately 9,000 years, from around 7050 BC to roughly 1500 AD. 41% of the tissue extracts tested positive for Trypanosoma cruzi kinetoplast DNA. 41%. Consistent across every cultural group. Every age bracket, both sexes. The parasite had been running at the same intensity for 9 millennia without interruption. That 41% prevalence rate matches modern endemic areas in the same region before public health interventions began. The disease has not changed. Separate studies confirmed the finding. Gould and colleagues in 1999 isolated T. cruzi DNA from 4,000-year-old mummified tissue stored at the Museo Arqueológico de San Pedro de Atacama. Ferreira and colleagues in 2000 confirmed the parasite in Atacama desert mummies dating from 2,000 years before present to 1400 AD. In one 15th to 16th century Peruvian mummy, the parasites themselves were identified by electron microscopy. The disease is called Chagas, named after Carlos Chagas, who first described it in 1909. The parasite is transmitted by triatomine insects, commonly known as kissing bugs. They feed on human blood at night. While feeding, they defecate near the bite wound. Trypanosoma cruzi enters the body through the wound or through mucous membranes. Once inside, it invades cardiac and smooth muscle cells. T. cruzi has co-evolved with armadillos, opossums, and other New World mammals for millions of years. The sylvatic cycle, the animal-to-animal transmission loop, was already well established when the first humans arrived on the Andean coast. Pre-Columbian mochi pottery from Peru, dating between roughly 100 and 800 AD, depicts facial swelling consistent with what is now called Romagna's sign. The disease was culturally recognized in ancient South America. What makes Chagas so dangerous is what it does after infection. The acute phase is often mild or completely asymptomatic. The patient may not know they are infected, but the parasite does not leave. It hides in the heart and gut tissue, sometimes for decades, before roughly 30% of patients develop severe cardiomyopathy or megacolon. The heart enlarges, it weakens, the gut stops functioning. There is no effective therapy for this chronic stage. Nifurtimox and benznidazole offer variable results in acute cases, but once the disease has settled in, treatment options are limited. The mechanism of chronic damage is still debated. The mainstream view is that the parasite persists directly in the tissues. PCR studies have found T. cruzi D NA inside damaged heart muscle. An alternative hypothesis proposes autoimmune damage, where the immune system, triggered by parasite proteins, begins attacking the heart's own tissue through antigenic mimicry. Scientific consensus has not been reached. For most of recorded medical history, the Western world had no idea Chagas existed. Carlos Chagas described it only in 1909, remarkably late for a disease affecting millions. It was invisible to European medicine because it existed only in the Americas and primarily affected rural, indigenous, and impoverished populations. The WHO eventually designated it a neglected tropical disease. Today, approximately 6 to 7 million people are infected worldwide. An estimated 300,000 cases exist in the United States alone. Tridamine bugs have been found across the American South, at least 11 species in Texas, Arizona, and New Mexico. Most Americans have never heard of a disease that exists in their own backyards. The WHO estimates Chagas kills roughly 12,000 people per year and represents the highest parasitic disease burden in the Americas by disability-adjusted life years. Same parasite, same vector, same 41% prevalence, running continuously from the chinchorro to the present day. And now, through globalization and migration, a 9,000-year-old problem is arriving in hospitals in cities that have never seen it before. But Chagas hides inside the body. The next disease on this list hides inside something even more fundamental. It hides inside the water. Number 3. Schistosomiasis, the snail-borne worm. The Nile gave Egypt its civilization. The annual floods deposited nutrient-rich silt across the floodplain, and the Egyptians built irrigation channels to control and distribute the water. Those channels also created something else: standing water. Warm, shallow, slow-moving pools, the perfect habitat for freshwater snails of the genus Malaria and Bulinus. Inside those snails lived the larvae of Schistosoma, parasitic blood flukes that would penetrate human skin on contact with water. The disease that built Egypt also built schistosomiasis. Calcified schistosoma eggs have been found in Egyptian mummies dating back 3,000 to 4,000 years. In 1910, Mark Armand Rufford discovered them in mummies from the 20th dynasty, roughly 1100 BC. It was one of the earliest paleoparasitological finds in history. More recently, Amber Campbell-Hibbs at Emory University detected Schistosoma mansoni proteins in the mummified skin of Nubian mummies from Sudan, approximately 1,500 years old. It was the first molecular proof of S. mansoni in any ancient civilization. The Wadi Halfa people who produced those mummies farmed the Nile using irrigation channels. 2 species of Schistosoma have dominated the human story, each adapted to a different ecological niche. E. s. hematobium, the urogenital form, thrives in moving, well-oxygenated water typical of the natural Nile. S. mansoni, the intestinal form, prefers stagnant, polluted water. As humans built more irrigation infrastructure, they created ideal conditions for S. mansoni to expand. The Ebers Papyrus, dating to roughly 1550 BC, describes blood in urine, a symptom widely interpreted as schistosomiasis. More than 3,000 years later, Napoleon's troops in Egypt between 1798 and 1801 suffered widespread hematuria from the same Nile canals. The life cycle is elegant and horrifying. Infected snails release free-swimming larvae, called cercariae, into the water. These larvae penetrate human skin during wading or swimming. Inside the body, the worms mature, pair up in the blood vessels, and begin producing eggs. Some of those eggs escape the body through urine or feces. Others become trapped in the liver, intestines, or bladder, provoking granulomatous inflammation that leads to fibrosis, portal hypertension, organ damage, and in the case of S. hematobium, bladder cancer. Ancient Egyptians clearly documented the symptom, blood in urine. But they never connected it to snails or worms. The full life cycle was not understood until Theodore Bilharz identified the parasite during a Cairo autopsy in 1851. 3,500 years of treating a symptom without understanding the cause. In the 1960s, the construction of the Aswan High Dam transformed schistosomiasis epidemiology in Egypt. The dam ended the annual Nile floods, replacing them with permanent irrigation channels. The snails thrived. Today, roughly 200 million people are infected with schistosomiasis worldwide. 700 million live in endemic areas. The disease kills an estimated 200,000 per year and is the second most devastating parasitic disease after malaria. It disproportionately affects children, causing anemia, stunted growth, and impaired cognitive development through repeated infection. Prehistoric, developed in the 1970s, remains the only widely available treatment. No vaccine exists. Drug resistance is emerging. The WHO's goal of eliminating schistosomiasis as a public health problem by 2030 is currently off track. Mass drug administration reaches only about 40% of those who need it, and the same engineering decisions that amplified the disease in ancient Egypt are being repeated today. Dam construction across Africa and Asia continues to create new standing water habitats for the snails. The life cycle has not changed in millennia. The same snail, the same worm, the same water. But Schistosomiasis is just one species of parasitic worm. The broader story of helminths reaches back not thousands, but hundreds of millions of years. Number 4. Parasitic worms. Helminths. In 2024, a study published in the journal Geology by Siheng Luo and colleagues at the Nanjing Institute of Geology and Paleontology described something remarkable. A 99-million-year-old tapeworm tentacle preserved in Burmese amber from the Kachin region of Myanmar. Using micro-CT scanning, the team revealed internal structures consistent with modern trypanorange tapeworms, the kind that parasitize sharks and rays. The specimen is the oldest body fossil of a tapeworm ever found. How a marine parasite ended up in tree resin is itself a story. The most likely scenario is that the host elasmobranch, a shark or ray, became stranded on a Cretaceous coastline. A land-based predator or scavenger tore into the carcass, and in the process, the tapeworm's tentacle was pulled free and landed in nearby resin, a freak event that preserved a parasite for nearly 100 million years. But the tapeworm is not the oldest evidence of helminth parasitism. Nematode eggs and what may be protozoan cysts have been found in a late Triassic phytosaur coprolite, more than 200 million years old. In May 2025, the Natural History Museum described Juracontocephalus, a 165-million-year-old thorny-headed worm from the Dauhugu Lagerstätte in Inner Mongolia, preserved in Jurassic volcanic lake sediment. It is the first body fossil of an acanthocephalin ever found, a group with over 1,400 modern species that infect fish, birds, and mammals. Devonian age fossils from Latvia, roughly 400 million years old, show circlets of hooks and sucking discs on fossil fish that are consistent with parasitic worm attachment structures. Helminth parasitism predates the dinosaurs. It predates flowering plants. It predates almost everything we think of as the modern natural world. The earliest known human parasite is a lung fluke. Its eggs found in fossilized coprolites from northern Chile, dating to approximately 5900 BC. Hookworm eggs have been identified in Brazilian coprolites from around 5000 BC. Roundworm eggs appear in Peruvian contexts from 2330 BC. Tapeworm eggs were found in Egyptian mummies from approximately 2000 BC, 1250 BC, and 1000 BC, including one mummy containing a well-preserved calcified female worm. The Ebers Papyrus references intestinal worms. Hippocrates documented them in fish, animals, and humans. The guinea worm, referenced in the Bible as fiery serpents, was preserved in Egyptian mummies. Helminths do not simply hide inside the body. They actively manipulate it. They secrete molecules that suppress the host immune response, dampening inflammation to avoid detection. They do not evade the immune system, they negotiate with it, and they can survive inside a human for years. Today, over 1.5 billion people carry soil-transmitted helminths, roughly 1 in 5 humans alive. Ascaris lumbricoides, the giant roundworm, infects approximately 800 million people. Adult worms reach 35 centimeters inside the small intestine. Whipworm infects roughly 500 million, embedding its thin anterior end into the intestinal wall to feed on blood. Hookworm alone causes an estimated 22 million disability-adjusted life years lost annually. There's one extraordinary success story. The guinea worm, which once infected 3.5 million people per year in 1986, was reduced to just 14 human cases in 2023. It is one of the most successful disease elimination campaigns in history. But here's the paradox. In wealthy nations where helminths have been eliminated, some people are now deliberately infecting themselves with parasitic worms to treat autoimmune conditions like Crohn's disease. The organisms we spent a century wiping out may turn out to be medically necessary. The Old Friends hypothesis proposes that the human immune system evolved alongside helminths, and that their absence in modern sanitized environments is contributing to the rise of autoimmune diseases. We eliminated worms from our bodies, but not from our immune system's expectations. More humans carry parasitic worms right now than any other disease in this video. The most common disease on Earth is also the most ignored, but the next entry does not hide, does not negotiate, and does not wait. It gets inside your brain, and once it does, nothing can save you. Number 5. Rabies, the oldest virus. Nearly 4,000 years ago in ancient Mesopotamia, a legal code was inscribed onto clay tablets. The Codex of Eshnunna, dating to roughly 1930 BC, included a provision that if a dog was known to be rabid and its owner failed to restrain it, and the dog then bit a person who died, the owner owed a fine of 40 shekels of silver. It is one of the oldest recorded references to any infectious disease. Aristotle described rabies in dogs around 350 BC and noted that the disease was transmitted through bites. The Roman physician Celsus, writing in the 1st century AD, recommended cauterizing bite wounds to prevent what he called hydrophobia. The word itself, rabies, comes from the Latin for madness, but the virus behind it is far older than any civilization. Rabies is caused by lyssaviruses, a family named from the Greek word lyssa, meaning rage. Phylogenetic analyses show that lyssaviruses co-evolved with bats. Bats are the ancestral reservoir for every known lyssavirus species. The diversification of lyssaviruses maps onto the diversification of bat lineages, suggesting the relationship stretches back tens of millions of years. No fossil evidence of the virus exists because viral RNA degrades far too quickly for preservation. But the molecular clock is clear. This virus was infecting mammals long before humans existed. What rabies does to the brain is extraordinary in its brutality. After entering through a bite wound, the virus travels along peripheral nerves toward the central nervous system at a rate of roughly 12 to 24 millimeters per day. The incubation period can last weeks, months, or in rare cases over a year, depending on how far the bite is from the brain. Once the virus reaches the brain, it causes acute viral encephalitis. The symptoms are unmistakable: hydrophobia, the violent spasming at the sight or sound of water; aerophobia, the fear of drafts; hyperactivity, confusion, aggression, paralysis. Death follows within days. There are 2 clinical forms. Furious rabies accounts for roughly 80% of cases and involves the agitation, aggression, and hydrophobia that have terrified humans for millennia. Paralytic rabies, the remaining 20%, involves progressive paralysis and is often misdiagnosed. Once clinical symptoms appear, rabies is nearly 100% fatal. It is one of the highest case fatality rates of any infectious disease known to medicine. The virus's strategy is devastatingly simple. By inducing aggression, it promotes biting, which transmits the virus to a new host. It is the same logic as the zombie fungus in section 1, but operating on vertebrates instead of insects. Evolution's most effective behavioral manipulation of a mammalian host. Rabies was essentially untreatable until 1885, when Louis Pasteur and Emile Roux developed the first vaccine, testing it on a 9-year-old boy named Joseph Meister, who survived. In 2004, the Milwaukee Protocol, involving induced coma and antiviral treatment, saved Gina Giese, a 15-year-old from Wisconsin, making her one of the only known unvaccinated survivors of symptomatic rabies. Subsequent attempts to replicate the protocol have largely failed. For millennia, rabies was not understood as a disease. It was understood as transformation, demonic possession, divine punishment. Some historians have proposed that rabies outbreaks in medieval Europe may have inspired werewolf legends. The progression from rational human to violent, frothing aggressor triggered by an animal bite maps closely onto the clinical presentation of furious rabies. The disease was managed through fear and folklore, not science. Today, rabies kills approximately 59,000 people per year. 95% of deaths occur in Africa and Asia. Dog bites account for up to 99% of human cases. India alone bears more than 20,000 deaths annually. Post-exposure prophylaxis is nearly 100% effective if administered before symptoms begin, but it costs $40 to $100 or more per course, which is prohibitive in many of the regions where rabies is most common. The WHO, OIE, and FAO have set a target of eliminating dog-mediated human rabies by 2030 through mass dog vaccination. The fundamental biology of rabies has not changed across millions of years, a virus that makes its host bite other hosts. The same mechanism, running since bats first diversified. But rabies destroys its victims in days. The next disease on this list works on a completely different timescale, disfiguring its victims over years while the world looks away. Number 6, leprosy. Hansen's disease. In 2025, a team led by Dario Ramirez at the National University of Córdoba in Argentina, working with Kirsten Boss at the Max Planck Institute, reconstructed 2 genomes of Mycobacterium lepromatosis from approximately 4,000-year-old human skeletons found in Chile. The finding proved that a form of leprosy existed in the Americas thousands of years before any European ship crossed the Atlantic. It overturned one of the longest-held assumptions in infectious disease history, that leprosy was brought to the New World by colonizers. M. lepromatosis and M. leprae, the two pathogens that cause Hansen's disease, evolved independently on opposite sides of the world. Two related organisms converging on the same devastating pathology from entirely different evolutionary starting points. Leprosy is one of the most stigmatized diseases in human history. Leper colonies, social exile, religious interpretations of divine punishment. At the peak of its medieval European prevalence, between the 10th and 14th centuries, more than 19,000 leprosaria existed across the continent. Skeletal evidence of leprosy spans 4,000 to 5,000 years in India, Egypt, and Europe. The oldest European case is a Celtic skeleton from Casalecchio di Reno, near Bologna, Italy, dating to the 4th or 3rd century BC. Ancient DNA phylogenetics suggest M. leprae itself originated approximately 7,000 years ago. The bacterium attacks peripheral nerves and skin. In its lepromatous form, it causes progressive destruction of the nasal septum, loss of eyebrows, skin nodules, and nerve damage that leads to insensitivity. Patients lose the ability to feel pain in their extremities, which leads to repeated unnoticed injuries and the disfigurement historically associated with the disease. M. leprae is an obligate human pathogen. It cannot survive outside the human body for extended periods. The only other known natural reservoir is the 9-banded armadillo in the southern United States. The incubation period averages 5 years but can exceed 20, making it one of the longest of any infectious disease. A remarkable discovery by Helen Donahue and Mark Spiegelman at University College London found both M. leprae and M. tuberculosis ancient DNA in a 1st century AD shrouded body from Jerusalem. The individual had likely been left unburied because of the stigma attached to leprosy. The co-infection finding overturned a long-standing theory about leprosy's medieval decline. The previous assumption was that tuberculosis provided cross-immunity against leprosy. The new evidence suggests something darker. TB killed leprosy patients before they could transmit leprosy to others. The decline was driven not by immunity but by death. Stigma suppressed scientific understanding of leprosy for centuries. It was treated as a moral or spiritual condition rather than a medical one. Patients were isolated rather than studied. The bacterium was not identified until Gerhard Armauer Hansen in 1873. Only about 5% of people are genetically susceptible to M. leprae. The majority of humans have natural resistance, yet the disease persists because it needs only a small vulnerable population to maintain transmission. Today, approximately 200,000 new cases are diagnosed annually. India, Brazil, and Indonesia account for more than 80%. Multi-drug therapy, introduced in the 1980s, cures leprosy. But nerve damage present at the time of diagnosis is irreversible, and diagnosis is frequently delayed because of stigma. The WHO declared leprosy eliminated as a public health problem in 2000, defined as fewer than 1 case per 10,000 people globally. But that statistical milestone masks ongoing transmission in endemic hotspots. In some countries, leprosy is still grounds for divorce. India repealed colonial-era laws that discriminated against people with leprosy only in 2019. The social disease outlives the medical one, and leprosy left something else behind. Selective pressure from M. leprae over thousands of years contributed to variants in the HLA gene complex that are still detectable in modern human populations. The disease literally wrote itself into human DNA, but leprosy targets the body from outside. The next entry attacks from within, and it has been doing so since before the dinosaurs went extinct. Number 7. Cancer, osteosarcoma. There is a persistent belief that cancer is a modern disease, that it is a product of processed food, industrial pollution, radiation, chemicals, stress, or some other feature of contemporary life. A 240 million-year-old turtle says otherwise. In the Triassic period, a shell-less stem turtle called Papochilus rossini developed a malignant bone tumor in its femur. The fossil was analyzed by Yara Harity and colleagues at the Museum für Naturkunde in Berlin using micro-CT scanning. Co-author Patrick Asbach, a radiologist at the Charité University of Medicine, noted that the tumor appeared almost exactly like osteosarcoma in humans, the same cancer, 240 million years apart. In 1989, a deformed leg bone was discovered in Dinosaur Provincial Park in Alberta, Canada. It belonged to a Centrosaurus apertus, a plant-eating horned dinosaur that lived 76 to 77 million years ago. For decades, scientists assumed the malformation was a healed fracture. In 2017, David Evans at the Royal Ontario Museum, along with pathologist Mark Crowther and osteopathologist Snezana Popovich at McMaster University, decided to re-examine the bone. They assembled a multidisciplinary team and used the same diagnostic process applied to human cancer patients: high-resolution CT scans, thin-section microscopy, three-dimensional reconstruction. The diagnosis, published in August 2020 in The Lancet Oncology, was osteosarcoma. It was the first time a malignant cancer had been confirmed at the cellular level in a dinosaur. The tumor was advanced. It may have metastasized. A human with an equivalent stage of the disease would likely not survive without treatment. But the Centrosaurus was found in a massive bone bed alongside hundreds of others, evidence of a herd killed by a flood. The researchers concluded that the herd's protection may have allowed this crippled individual to survive longer than it would have alone. The earliest known cancer in a human ancestor is also osteosarcoma, identified in a 1.7 million year old hominin metatarsal from Swartkrans Cave in South Africa. Another dinosaur, Telmatosaurus transylvanicus from the Haiteg Basin in Romania, 66 to 70 million years old, showed a possible amyloblastoma in its jaw. A 2025 study from Anglia Ruskin University also found structures resembling preserved red blood cells in the tumor tissue. The connection between cancer and rapid growth is significant. Dinosaurs grew at extraordinary rates from birth. Modern dog breeds that grow quickly, like Great Danes and Rottweilers, have elevated osteosarcoma risk. In humans, osteosarcoma primarily strikes adolescents during growth spurts. The reason the cancer's modern myth persists is simple. In the past, most people did not live long enough to develop the cancers that dominate modern mortality statistics, and soft tissue cancers almost never fossilize. The absence of evidence was mistaken for evidence of absence. Cancer is not something civilization gave us, it is something multicellular life was born with. The vulnerability is built into the mechanism of cell division itself. Any organism with dividing cells can develop cancer. The WHO estimates that cancer killed 9.7 million people in 2022. Osteosarcoma in humans accounts for roughly 800 to 900 new cases per year in the United States, primarily in children and young adults. The 5-year survival rate for localized cases is about 65%. For metastatic cases, it drops to roughly 20%. Those numbers have not improved in 35 years. There's an evolutionary puzzle here called Peto's paradox. Large animals have more cells, which means more opportunities for cancerous mutations. Yet elephants, which carry extra copies of the TP53 tumor suppressor gene, and bowhead whales, which have ultra-efficient DNA repair mechanisms, get cancer at lower rates than expected. Dinosaurs, some of the largest animals to ever exist, may have possessed similar mechanisms that we have not yet discovered. Preserved soft tissue proteins in dinosaur fossils could potentially reveal ancient tumor suppression strategies. The 2025 Anglia Ruskin study explicitly called for better fossil conservation protocols to enable this kind of molecular research. Paleontology is positioning itself as a tool for human medicine. Understanding why cancer has been running the same biological program for 240 million years may finally explain why it remains so resistant to treatment. But cancer is embedded in the code of life itself. The next disease is an outsider, a parasite that invaded from another species and then rewrote the human genome through sheer killing power. Number 8, malaria. No disease in history has exerted more evolutionary pressure on the human genome than malaria. Sickle cell trait, G6PD deficiency, thalassemia, Duffy negative blood type. These are not random mutations, they are genetic scars, each one providing partial resistance to the Plasmodium parasite, each one selected for over thousands of generations because the people who carried them were slightly more likely to survive malaria and reproduce. Collectively, these mutations affect hundreds of millions of people alive today. Many of them cause serious medical conditions of their own. Sickle cell disease is painful and often fatal. Thalassemia requires lifelong blood transfusions. G. 6PD deficiency triggers hemolytic crises. They exist anyway, because malaria is worse. This is the strongest evidence of natural selection operating on the modern human genome. Malaria has literally reshaped human DNA. The Plasmodium parasite family is far older than humans. It has co-evolved with primates for millions of years. Bird malaria lineages diverged from mammalian lineages tens of millions of years ago. Ancient mosquitoes preserved in amber show blood-feeding morphology as far back as the Cretaceous. The mosquito-parasite vertebrate transmission triangle has deep evolutionary roots. The deadliest human malaria, caused by Plasmodium falciparum, likely jumped from gorillas to humans within the last 10 to 50,000 years, based on genomic analyses published by Wyman Liu and colleagues in Nature in 2010. Plasmodium vivax, which causes a less lethal but more widespread form of the disease, is older and may predate the out-of-Africa migration. Its ability to lie dormant in the liver as a hypnozoite suggests a long period of adaptation to human physiology. Plasmodium DNA has been detected in Egyptian mummies. A study by Andreas Nerlich and colleagues in 2008 identified P. falciparum DNA in mummy tissue roughly 3,200 years old. In 2010, Zahi Hawass and colleagues found Plasmodium DNA in the mummy of Tutankhamun, dating to approximately 1324 BC. CT scans of the pharaoh's body revealed bone necrosis consistent with a vascular bone disease. Malaria may have contributed to his death. Hippocrates described quartan and tertian fevers that matched the 72-hour and 48-hour cycles of different Plasmodium species. He associated them with swampy environments. The Romans called the disease malaria, bad air, believing that the foul vapors from marshes were the cause. They were right about the geography but wrong about the mechanism. The mosquito vector was not identified until Ronald Ross's experiments in 1897. For thousands of years, humans correctly linked the disease to wetlands but attributed causation to invisible miasma rather than the insects breeding in the water. Today, malaria kills roughly 608,000 people per year. 95% of deaths occur in sub-Saharan Africa. Children under 5 account for approximately 80% of those deaths. Roughly 1 child dies from malaria every minute. The first malaria vaccines have finally arrived after decades of failure. RTSS, developed by GSK and known as Musquirix, was recommended by the WHO in 2021. R21/Matrix-M, developed at the University of Oxford, was recommended in 2023 with higher efficacy of approximately 75%. But artemisinin resistance, confirmed in Southeast Asia and now emerging in parts of Africa, threatens the entire treatment framework. Insecticide-treated bed nets reduced malaria deaths by roughly 50% between 2000 and 2015, but progress has stalled as mosquito resistance to pyrethroids rises. Climate change is expanding the range of Anopheles mosquitoes to higher altitudes and latitudes. Malaria transmission is now occurring in highland areas of East Africa, where it was historically absent. No tropical endemic zone has ever been permanently cleared of malaria, despite billions of dollars in investment. Malaria has killed more humans than any other infectious disease in history, and it shows no sign of letting go. But malaria reshapes the genome slowly, over tens of thousands of years. The next disease on this list reshaped history in a matter of months, and it started as a harmless gut bacterium. Number 9, Yersinia pestis, the plague. 5,000 years ago, a man in his 20s or 30s was buried near the banks of a river in what is now Latvia. His name is lost. Researchers call him REV 2039. In 2021, a team led by Ben Crosicura at the University of Kiel published a study in Cell Reports revealing that ancient DNA and proteins extracted from this man's bones and teeth contained Yersinia pestis. The plague bacterium was circulating in his bloodstream when he died. He is the oldest confirmed plague victim in the archaeological record. But the strain he carried was not the plague that would later destroy civilizations. It was the most basal lineage ever found, the closest known relative to the common ancestor of all Y. pestis. It lacked the key virulence genes required for flea-borne transmission. It could not cause bubonic plague. RV 2039 was probably bitten by an infected rodent. He likely developed septicemia and died within days. He was carefully buried. No mass graves surrounded him. This was not an epidemic. It was a single unfortunate encounter between a human and a bacterium that was still learning how to kill. Yersinia pestis evolved from Yersinia pseudotuberculosis, a relatively harmless gut bacterium. Genomic analysis suggests the split occurred between 5,700 and 7,000 years ago. What followed was one of the fastest and most lethal evolutionary transformations in pathogen history. The bacterium acquired the T gene, which allowed it to survive inside the flea gut. It lost its flagellum, reducing detection by the host immune system. It gained the PPCP1 plasmid, enhancing its ability to invade tissue. Step by step, a gut bacterium became a flea-vectored systemic killer. In 2018, Nicholas Raskovan and colleagues published a study in Cell that found Y. pestis in a 20-year-old funnel beaker woman from Sweden. She had no Yamnaya ancestry, proving that plague existed in Europe before the steppe migrations that some researchers had credited with bringing the disease westward. That same year, Maria Spiro and colleagues published in Nature Communications the reconstruction of Y. pestis genomes roughly 3,800 years old that carried the YMT gene and other flea adaptation markers. It was the first genomic evidence that bubonic plague was possible by the late Bronze Age. In 2023, Swali and colleagues sequenced 3 Y. pestis genomes from Britain, all approximately 4,000 years old, from Charterhouse Warren in Somerset and Levains in Cumbria, the oldest plague in Britain. 3 historically documented pandemics followed. The plague of Justinian, beginning in 541 AD and lasting intermittently until roughly 750 AD, confirmed in 2025 through ancient DNA from a mass grave in Jerash, Jordan. The Black Death, which killed an estimated 30 to 60% of Europe's population between 1347 and 1353. And the third pandemic, which emerged in China in the 1850s and spread globally. For centuries, plague was attributed to divine wrath, astrological alignments, poisoned wells, and miasma. The bacterium was not identified until Alexandre Yersin isolated it during the Hong Kong outbreak in 1894. The flea vector was not confirmed until Paul Louis Simon's experiments in 1898. The entire mechanical linkage between rats, fleas, and humans was invisible for the duration of the first 2 pandemics. A 2022 study by Jennifer Klunk and colleagues published in Nature demonstrated that 4 specific immune gene variants were under strong positive selection during and after the Black Death. The people who survived carried genetic variants that enhanced their immune response to Y. pestis, but those same variants are now associated with increased susceptibility to autoimmune diseases, including Crohn's disease and rheumatoid arthritis. The Black Death permanently altered the European immune system. We are living with the autoimmune consequences. Today, plague has not disappeared. Roughly 1 to 2,000 cases are reported to the WHO annually. Madagascar experienced an outbreak in 2017 with over 2,400 suspected cases and 209 deaths, including pneumonic plague in the capital Antananarivo. In the United States, approximately 7 cases occur per year, predominantly in New Mexico, Arizona, and Colorado. Plague is treatable with antibiotics if caught early, but untreated bubonic plague has a fatality rate of 30 to 60%, and untreated pneumonic plague is nearly 100% fatal. Antibiotic-resistant strains have already been documented. A multi-drug-resistant Y. pestis was isolated in Madagascar in 1995. The CDC classifies Y. pestis as a Tier 1 select agent because of its potential as a bioweapon. Sylvatic reservoirs in rodent populations exist on every continent except Australia and Antarctica. Plague can never be eradicated. The rodents are still carrying it. The fleas are still biting. The bacterium is still evolving. From harmless gut bacterium to civilization ender in roughly 4,000 years of evolution. But there is one disease that makes even plague look young. It has been with us for 70,000 years. It was with the Neanderthals before us. And it is still the leading infectious killer on Earth. Number 10, tuberculosis. In a cave called Sobolyuk in the Buk Mountains of Hungary, the remains of 2 Neanderthals were found, an adult and a child. György Palfer and colleagues at the University of Szeged identified lytic lesions along the adult's spine and on the interior of the child's skull, bone destruction patterns consistent with tuberculosis. Una Lee at the University of Birmingham then extracted and analyzed samples from both skeletons and confirmed the presence of Mycobacterium tuberculosis, DNA. It was the first confirmed case of tuberculosis in a non-human hominin species. The Neanderthals likely contracted TB from hunting and butchering infected bison. Large European prey animals, particularly bison, show evidence of tuberculosis in the archaeological record. The disease posed a double threat. It could infect the hunters directly, and it could decimate the prey populations they depended on. Some researchers have proposed that tuberculosis may have contributed to Neanderthal extinction. A species that had survived ice ages could not survive a bacterium. Molecular clock analyses placed the Mycobacterium tuberculosis complex in human populations of Africa for at least 70,000 years. It expanded alongside the out-of-Africa migration, diversifying as human populations spread across the globe. The most virulent and drug-resistant modern lineage, the so-called Beijing strains or lineage 2, traces its expansion to the arrival of anatomically modern humans in East Asia between 42,000 and 32,000 years ago, with later pulses of expansion linked to the development of agriculture in China roughly 8,000 years ago. The earliest skeletal case confirmed by ancient DNA comes from a submerged village. Atlat Yam was a pre-pottery Neolithic settlement on the coast of what is now Haifa, Israel, occupied roughly 9,000 years ago. It is now underwater. Israel Hershkovitz at Tel Aviv University noticed bone lesions consistent with TB in two skeletons from the site, a woman and an infant. Helen Donahue and Mark Spiegelman at University College London recovered M. tuberculosis, DNA, and cell wall lipids from the bones. The village had domesticated cattle. The obvious assumption was that the humans had caught TB from the animals. But the molecular analysis proved otherwise. The strain was definitively human, not bovine. Mycobacterium tuberculosis predates Mycobacterium bovis. TB has a human origin, not a cattle origin. That finding overturned a theory that had dominated the field for decades, the traditional view that humans acquired tuberculosis from newly domesticated cattle during the Neolithic Revolution. The Atlat Yam strain had already lost a specific genetic marker characteristic of a major modern strain family, showing that the bacterium was already diversifying 9,000 years ago. What makes tuberculosis so extraordinarily successful is what it does inside the body. M. tuberculosis is inhaled into the lungs where it is engulfed by macrophages, the immune cells whose entire purpose is to destroy invading pathogens. But M. tuberculosis does not die inside the macrophage. It blocks the fusion of the phagosome with the lysosome, the mechanism by which the cell would normally digest and destroy the invader. The bacterium survives inside the very cell sent to kill it. And it can remain there, dormant, for decades. This is latent tuberculosis. 1/4 of the entire human population, roughly 2 billion people, carry latent M. tuberculosis right now. Most will never develop active disease, but the bacterium is waiting. The hallmark skeletal sign of TB is Pott's disease, spinal tuberculosis, where the bacterium destroys vertebral bodies, causing them to collapse and creating the angular deformity that paleopathologists can identify in ancient remains from every inhabited continent. Tuberculosis thrived in cities. During the Industrial Revolution, crowded tenement housing, poor sanitation, and widespread malnutrition created the ideal conditions for airborne transmission. TB was known as the White Plague. It killed 1 in 4 Europeans in the 19th century. Robert Koch identified M. tuberculosis as the causative agent on March 24, 1882. The date is now commemorated as World TB Day. The BCG vaccine, developed in 1921, remains the only licensed TB vaccine in the world. It is over 100 years old. Its protection against pulmonary TB in adults ranges from 0 to 80%, depending on the population. No replacement is currently in widespread use. In 2022, tuberculosis killed approximately 1.3 million people, making it the leading cause of death from a single infectious agent on Earth. Roughly 10.6 million people developed active TB disease that year. Multi-drug resistant TB, resistant to at least isoniazid and rifampicin, accounts for approximately 450,000 new cases annually. Treatment for drug-susceptible TB still requires 4 to 6 months of daily multi-drug therapy. MDR TB treatment can last 9 to 20 months using more toxic drugs with cure rates of only about 60%. Extensively, drug-resistant TB is even harder to treat. 8 countries account for 2/3 of all TB cases worldwide: India, Indonesia, China, the Philippines, Pakistan, Nigeria, Bangladesh, and the Democratic Republic of Congo. After 70,000 years of coevolution with humans, Mycobacterium tuberculosis has developed strategies for immune evasion that no other pathogen can match. It survives inside the cells designed to destroy it. It can remain dormant for a human lifetime. It has more human hosts now than at any point in its history, and it is developing resistance to every drug we have built to fight it. It is, by any reasonable measure, the single most successful human pathogen in evolutionary history. The diseases in this video did not just kill people, they reshaped the human species at the genetic level. Malaria forced sickle cell into existence, plague selected for immune variants that now drive Crohn's disease and rheumatoid arthritis. Leprosy carved itself into the HLA gene complex. Tuberculosis achieved détente, sitting dormant inside the immune cells meant to destroy it, latent in 1/4 of all humans alive. And the worms. The old friends hypothesis proposes that our immune systems were calibrated by parasites we have since eliminated. Without them, the system misfires. Autoimmune disease may be the cost of modern hygiene. Meanwhile, drug resistance is accelerating across every disease in this video. Artemisinin-resistant malaria, extensively drug-resistant tuberculosis, antifungal-resistant Candida auris. Climate change is expanding vector ranges into temperate zones. The geographic containment wealthy nations relied on is failing. Every disease in this video shapes something about what it means to be human. We built irrigation and created Schistosomiasis, domesticated dogs and spread rabies, settled cities and amplified plague. The relationship is not predator and prey, it is co-creation. Every disease in this video was here before us. Everyone will be here after us. The oldest is 240 million years old. The newest drug we have to fight it is 35 years old. The math is not in our favor.